Welcome to G Fun Facts Online. For a long time, we thought of our DNA as an unchangeable blueprint, a fixed script that determined our destiny from eye color to disease risk. But what if I told you that this script has a conductor? A conductor that can choose which parts of the music are played loudly, which are played softly, and which are silenced altogether. And what if I told you that conductor is the world around you? This is the revolutionary field of environmental epigenetics, which reveals a dynamic conversation between your genes and your environment, showing how the food you eat, the air you breathe, and even the experiences you have can rewrite your biological story without changing a single letter of your DNA. So how does this work? To understand the power of epigenetics, we first need to distinguish it from traditional genetics. Think of your genome as a massive cookbook. Genetics is the study of the recipes themselves, the sequence of ingredients and instructions written in the book. A genetic mutation is like permanently rewriting one of those recipes. Epigenetics, on the other hand, means above genetics. It's not about the recipes but how they're used. It's the collection of sticky notes, highlights, and annotations that a chef, representing your environment, adds to the cookbook. These marks don't change the recipe's text, but they absolutely influence which meals get cooked. This is the key to understanding one of biology's greatest puzzles. Why is a heart cell so different from a brain cell when they both contain the exact same set of DNA, the same cookbook? The answer is epigenetics. During development, epigenetic marks selectively activate the heart cell recipes in one cell while silencing them in another, instead highlighting the brain cell recipes. Environmental epigenetics shows us that this process isn't just for early development. It's happening throughout our entire lives, providing a molecular link between our genes and our world. So how exactly does the environment leave these sticky notes on our DNA? It uses a sophisticated molecular toolkit with three main mechanisms. First up is DNA methylation, which acts like an off switch. This process involves adding a tiny chemical tag called a methyl group directly onto the DNA molecule itself, usually at a specific spot. Think of these methyl groups as locks placed on a gene. When a gene gets covered in these locks, the cell's machinery can't access it to read its instructions. The gene is effectively silenced, turned off. Environmental factors are known to directly influence these methylation patterns. For example, your diet is crucial. Nutrients like folate and B vitamins, found in leafy greens, are essential ingredients for making these methyl groups. A poor diet can lead to improper gene locking, potentially activating genes that should remain silent. Toxins are another major factor. Pollutants like heavy metals and benzene can cause widespread, harmful changes to these methylation locks. Even chronic stress can trigger hormonal changes that alter the methylation of genes involved in our stress response, potentially contributing to conditions like depression. The second tool is histone modification, which works more like a volume control dial. Our incredibly long DNA strands aren't just floating around in our cells. They're tightly wound around proteins called histones, like thread around a spool. This packaging structure is called chromatin. Histone modification involves adding or removing chemical tags to the tails of these histone proteins. One common modification is acetylation. Adding an acetyl group generally loosens the chromatin, like unspooling the thread, making the genes on that section of DNA more accessible and turning their expression volume up. Histone methylation is more complex. Depending on where the methyl tag is placed, it can either turn the volume up or down. The enzymes that control these tags are highly sensitive to environmental signals. For instance, a compound called sulforaphane, found in broccoli, can block the enzymes that remove those volume-up acetyl tags. This can lead to the activation of beneficial disease-preventing genes. So yes, your mom was right. Eating your broccoli can literally change your gene expression for the better. The third mechanism involves a class of molecules called non-coding RNAs, which act as fine-tuning regulators. For decades, scientists thought the vast stretches of our DNA that don't code for proteins were junk DNA. We now know this couldn't be further from the truth. This DNA produces a diverse family of non-coding RNA molecules. One key type is micro-RNAs. These are tiny RNA molecules that can intercept the message from a gene before it gets turned into a protein. They can block the message or mark it for destruction, effectively silencing a gene after its initial instructions have been read. Their expression is highly sensitive to stressors like UV radiation and pollutants, and when they're dysregulated, they can disrupt the delicate balance of our cells, contributing to diseases like cancer. Together, DNA methylation, histone modification, and non-coding RNAs form an intricate network, a molecular language through which our environment constantly speaks to our genes. These concepts come to life in some truly incredible real-world examples. Perhaps the most famous is the Agouti mouse experiment. These mice carry a gene called Agouti, which, when active, gives them a yellow coat and makes them prone to obesity and diabetes. In a landmark study, scientists fed pregnant Agouti mice a diet rich in methyl donors, the same nutrients like folic acid and vitamin B12 we mentioned earlier. The pups born to these mothers were genetically identical to other Agouti mice, but they looked completely different. They were slim, had brown coats, and were perfectly healthy. The methyl-rich diet had provided the building blocks to place off switches on the Agouti gene, silencing it during development. A simple dietary change in the mother created a profoundly different health outcome in her offspring. The environment isn't just about what we eat. It's also our social world. Studies in rats showed that the amount of licking and grooming a mother gives her pup in its first week of life permanently shapes its response to stress. Pups that receive lots of nurturing grow up to be calm adults with a moderate stress response. Pups who are neglected grow up to be fearful and have an exaggerated stress response. The difference was traced to an epigenetic mark in the brain. The nurtured pups had the methyl locks removed from their glucocorticoid receptor gene, a gene that helps manage stress hormones. This made the gene more active, allowing them to handle stress better. The neglected pups kept the gene locked and silenced, leading to a lifetime of heightened anxiety. One of the most tragic and compelling human examples is the Dutch hunger winter of 1944. During a Nazi blockade, people in the Netherlands experienced severe famine. Decades later, scientists studied the children of women who were pregnant during this time. They found these individuals had significantly higher rates of obesity, diabetes, and heart disease in adulthood compared to their unexposed siblings. Epigenetic analysis revealed that the famine had left a permanent mark, altering the methylation on genes involved in metabolism. Their bodies were epigenetically programmed in the womb to survive a famine, a trait that became a major health liability in a world of plentiful food. This brings us to one of the most mind-bending ideas in this field: transgenerational epigenetic inheritance. This is the concept that the epigenetic marks from an individual's experiences might be passed down to their children, and even grandchildren. For this to happen, the marks must somehow survive a massive reprogramming event that erases most epigenetic tags during the formation of sperm and eggs. But it seems some marks can escape this erasure. Animal studies provide stunning evidence. In one experiment, male mice were trained to fear the smell of cherry blossoms by pairing it with a mild shock. Astonishingly, their children, and even their grandchildren, also showed a fear of that specific smell, despite never having encountered it before. An analysis of the father's sperm revealed that the gene for that specific odor receptor had been epigenetically altered. The father's learned experience was passed down through his germline. These discoveries have profound implications for our understanding of health and disease. Cancer, for example, is now seen as much a disease of the epigenome as it is of the genome. Many cancers feature epigenetic chaos, where tumor suppressor genes, the guardians of the cell, are silenced by methylation, while genes that promote growth run wild. Likewise, the brain is incredibly sensitive to environmental cues. Chronic stress and early life trauma can embed themselves as epigenetic memories, altering the regulation of stress response genes and increasing susceptibility to depression and anxiety. The good news is that because epigenetic marks are annotations, not permanent changes to the DNA sequence, they are potentially reversible. This has opened up an entirely new frontier in medicine. Scientists are developing epi-drugs that aim to correct faulty epigenetic patterns. For instance, drugs that inhibit DNA methylation can be used to reawaken silenced tumor suppressor genes in cancer cells, restoring the body's natural defenses. But beyond pharmaceuticals, there is immense power in lifestyle. Can we design diets or exercise plans to beneficially modify our epigenomes? The research strongly suggests yes. This moves us away from the old, rigid idea of genetic determinism and toward a more fluid and empowering reality. Our DNA is not our destiny. It is a dynamic script, and our life experiences are constantly interpreting and annotating it. The story of epigenetics reveals that the food we eat, the stress we endure, and the care we receive are woven into the very fabric of our being. We are not just passive inheritors of a fixed genetic code. We are active participants in the ongoing creation of our own biology. By understanding this remarkable molecular language, we are empowered with the knowledge to make choices that can positively rewrite our own genetic expression for a healthier life, and potentially a healthier future for generations to come. Thanks for listening to G Fun Facts Online.